Thank you everyone for joining us for another edition of Wildlife Wednesday at the Center for Wildlife Studies. My name is Jeff Stetz. I'm the Director of Science and an assistant professor here at the center. We're also joined again this week or this month by the president of CWS, Jack Hopkins, who is visiting me in Alaska currently, along with some other uh, very fortunate activities. There he is.
Hi
Sorry,
all.
we have folks still coming in, so I may be distracted a little bit by letting people in. Uh, so this program is something we put on every month, typically the second Wednesday of every month, where it's a chance for us to showcase the work of our collaborators, instructors, consultants, and sometimes just folks who we know of who are doing some great work in the conservation world. So we really do like to promote the work and conservation activities of our colleagues. And today is a great example of that. Very excited. Take a little deviation from our somewhat bear heavy program lineup to talk about things that matter to all animals, us included, about the adaptations of plants to extreme environments. As far as introduction, Dr. Doug here is a plant biologist, toxicologist, and educator whose research explores how environmental and human-induced stressors affect ecosystems and how plants adapt to challenging conditions at the molecular level. He spent 15 years as a professor of plant biology at Coastal Carolina University, where he studied plant responses to heavy metals and nutrient deprivation. He now serves as a toxicologist with the Maine Department of Agriculture, evaluating and communicating the risks of pesticides to human health and the environment. It's a very common topic yesterday among those of us on the river. Uh, and Doug just so happens to teach plant ecophysiology here at the Center for Wildlife Studies, which is starting here in early September. And that course focuses on connecting fundamental plant ecology with practical conservation and highlighting the essential role of healthy plants in supporting wildlife and functioning ecosystems. And with that, I am very happy to turn this over to Doug. If folks have questions during the talk, I guess it's often nice to maybe hold those off until the end. But if you want something addressed, and if Doug wants to field those mid-presentation, that's totally up to him, of course. But there will be a dedicated question and answer period at the end of this. And also, if you do have to jump off the presentation early, we do post these to a page on YouTube, and I'll share some of these links, like for Doug's course and for the playlist on YouTube a little bit later on in the presentation. And we always encourage folks to share that with their colleagues as well. And with that, I am super happy to turn this over to Dr. Doug and look forward to learning about these extreme adaptations.
Thanks, Jeff. I appreciate your kind introduction. I appreciate you inviting me to participate in Wildlife Wednesday. And as Jeff had said, this if you have questions, you're welcome to fire away during this discussion, or alternatively, you can wait to the end. Um, Yeah, Jeff mentioned that, I was a professor for 15 years at Coastal Carolina University, formerly trained as a molecular and cellular biologist, chose to study plants. But even though I lived in the world of molecular cellular biology, I always framed my questions in an ecological context, right? So I always wanted to think about the physiological ramifications of my studies, often thinking about how my research program could be applied to improving crop development, but also just wanting to learn, some basic scientific questions that might not have any immediate societal ramifications, and just wanting to learn about how plants grow in their natural environment. And I'm fascinated by this topic because plants, in contrast to animals, have different ways of adapting to stress. This is not to say that animals don't have to confront and adapt to stress or respond to stress, but they do so differently, right? And this is the one of the hallmarks of biology, that all organisms encounter stress. They are subjected to these environmental insults, and they must respond in an adequate way. For today's talk, we're going to define stress as any type of uh, constraint, any type of environmental factor that impairs the growth, the physiology, or the reproductive success of that species. Um, and this could be many different types of stress, right? You could think of heat stress, cold stress. We could think of nutrient deprivation, metal stress, salt stress are some of the common threats that might decrease the physiological performance of plants. Um, when we think of animals, including humans, right, we encounter stress, but we can sometimes mitigate that stress by moving to a different environment, right? So I use this picture on this left of this, this really cute furry golden retriever, right, on the beach. It's too hot. It doesn't like where it is. What's it do? So it just wanders off and seeks some shade, and all is good, right? The body temperature drops down to a homeostatic level. It's no longer panting or sweating. It's conserving water, and look at the dog. It's yeah, super happy and content. But plants are literally rooted in the soil. Typically, not all plants have to have roots or embed in the soil, but certainly majority are. And they cannot just simply move to a better location. They can't simply move to a location which lacks herbivores, or maybe a location where the nutrient status is more to their liking, or seek a different environment where the soil holds more water, or maybe the soil has less salt. Um, but and because of that, because plants can't just simply move locations, they have evolved a wide variety of adaptations and ways to respond to stress that helps them survive. So I use this, image of a tomato plant on the right. This is a common feature, in my backyard right now. If I don't water my plants daily, my plants are going to wilt. I suspect that the fresh weight of this tomato plant might fluctuate by 20 to 40% during the day if it's not wilted. So in other words, if it loses a drastic amount of water, it can rapidly lose all that weight, and yet it can bounce back, right? And this is something that we commonly observe. Certainly this couldn't happen to humans, right? I mean, we can't lose like 20, 30% of our body weight in a day and then gain it back, and everything's fine, right? But this tomato plant, you can water it, and in half an hour it's going to bounce back and look totally fine. Um, the wilting of these leaves is just one way that this tomato plant has adapted to water loss. And there are a variety of ways that we see plants adapting to stress in their environment. We know that cacti don't have true leaves to help them conserve water in really hot, arid environments. Um, when we think about plant stress, we can divide this into two different camps, two different flavors. We can think of both biotic stress and abiotic stress. My research program really focused on abiotic stress, and therefore that's going to really be the focus of this discussion tonight. But I just wanted to mention again that plants do encounter biotic stress, stress imposed by microbes such as bacteria, fungi, stress imposed by viruses, non-living entities that can infect plants and decrease their physiology and performance, as well as insects and animals. And in response to all these biotic threats, plants have evolved this wonderful arsenal of secondary metabolites that aren't necessarily required for growth under optimal conditions. But unless you're growing plants in a controlled laboratory setting, the plant's never going to encounter an ideal environment, right? And so because of that, plants again have evolved this arsenal of compounds that can help deter herbivores. So shown here are some common molecules that we're aware of. Some have medicinal value. Some are abused recreationally. But I illustrate this just to show that plants have adapted to biotic stress by evolving these really wild compounds that can interfere with the physiology of insects and animals. I suppose this also has applications to my current job. Jeff mentioned that I am currently a toxicologist for the Maine Board of Pesticide Control, which is under the Department of Agriculture. And so part of my current position is evaluating the risks of pesticides as they pertain to human health and the environment. All right. Um, so what I thought would be, what I hope to be beneficial during our limited time here is just to discuss three different types of stress that plants encounter and how plants adapt to this stress. Um, so the first is adjustments to photosynthesis. So light intensity can vary, and based on the plant's environment, it can respond differently to different intensities of light. So on the top left, we have a picture of the seedlings moving towards the light. This is a common phenomenon, and I suspect that people have seen this as they put their plants near windows, and sometimes those flowers and leaves will gravitate towards the light. This is called heliotropism. The root of the word helio suggests sun. Tropism means movement, so it really just means movement towards sun. So these plants are being limited by light, and in response, they're going to adjust their growth and move towards that light. And this type of growth is mediated by a hormone called auxin. Um, Then shown on the bottom is a different, perhaps less obvious way of how plants can respond to light limitations. So shown here are two different chloroplasts. So the outline of the chloroplast is, well, depicted by this membrane. Um, and inside these chloroplasts are stacked thylakoids. And for those who remember, introductory biology, these stacked thylakoids are where the light reactions occur. So these are the structures that absorb light energy, and that light energy will ultimately be converted into sugars in the stroma of the chloroplasts. So I want you to look at this slide on the bottom, or this figure in the bottom right. You can see that these two different chloroplasts have different thylakoid abundance and structure. And so one of these chloroplasts is being deprived by light. Um, if we were together, I would ask you, which one do you think it is? Is it the chloroplast on the left that's deprived of light, or the chloroplast on the right that is being deprived of light? And so we can certainly see that there are more thylakoids um, uh, in the chloroplast on the right. And so, in fact, this chloroplast on the right is from a plant that's being deprived by light. So that's another sub. This would be a subcellular response. In response to light limitations, the number of thylakoids in a chloroplast can increase. And not only that, but these thylakoids and the chloroplasts can move within a cell. So you can see how these chloroplasts can within a cell, these chloroplasts can move towards the light in hopes of capturing as much light energy as possible. So that's another example of how plants can adapt to light limitation. We often think of plants as always being, perhaps craving light and being able to handle as much light as possible. But that's not the case, right? So sometimes too much light is toxic to the plants. We know this because there are indoor plants, and you can't just take your indoor plant and move it outside. This is also true for plants that have been acclimated to indoor lighting, such as a lime tree. I remember the what happened when I had my lime tree indoors all winter, and then I tried to gradually, let it handle some increasing light. But then one day it just was like way sunny, the lime tree was toast, right? So even though lime trees, of course, in nature they're grown outdoors where they can handle a lot of light, but I just roasted my lime tree because it had been inside the house all winter, experiencing low light intensity. All of a sudden, was blasted by high light intensity, and I fried the plant. But to take this in a more natural setting, I suppose, we're going to examine this evergreen herb called Gallix ursulata on the top left. So this is a plant that is found from northeast United States all the way through the eastern seaboard. So it has a broad geographic range, but this is a plant that's found in the understory, right? And so typically it does not get, typically it is not going to receive a lot of light because there is a canopy, which is going to filter out that light. However, there are times perhaps when there's a clearing in the forest and all of a sudden these plants are going to be exposed to a lot more light, higher light intensity than they're used to, right? And so these plants are now being challenged by too much light, right? So too much light for these plants is toxic, right? So these are not plants that are used to or have evolved to grow in lots of light. So one metabolic response that they can, I guess, execute, or one way that they can respond is by increasing the amount of a molecule called anthocyanin. Anthocyanin is a pigment, but it's not a pigment that participates in photosynthesis like chlorophyll. Um, can be explained because this pigment is found in the vacuole of plant cells. And because it's found in the vacuole, which is a aqueous solution containing salt, sugars, osmolytes, and, well, in this case, a pigment, it does not participate in photosynthesis. So this pigment, when you isolate it, it's reddish or pinkish. This pigment is going to act as a way to dissipate extra energy. I suppose it might be inappropriate to make the analogy that this acts as sunscreen, but if you have that kind of concept, then I think that kind of works, right? It's a way the anthocyanin will accumulate in these plants, and the purpose is to absorb that extra sunlight that the plant does not want. This is because extra sunlight's toxic. So when that sun energy, we know that the photons have a lot of energy, right? And we know this because we can collect solar energy from panels, right, and drive our electronics. So the sun has a whole bunch of energy. And if that energy cannot be handled, that energy from the sun cannot be tightly controlled and regulated in that chloroplasts, it's going to cause just absolute havoc in the chloroplasts and in any type of photosynthetic material. There will be a whole bunch of reactive oxygen species that's generated if the plant does not have a way to dissipate or perhaps handle that excessive energy. So in this case, the anthocyanin accumulates as a way to absorb that extra energy to try to alleviate the chloroplasts from handling too much energy. Okay. Um, so another example of ways that plants can absorb excessive energy comes from evergreens, right? So here's a conifer. In the background, you can see that there are some deciduous trees that have appropriately dropped their leaves in the winter months. This is a strategic evolutionary programmed response, right? Because these broadleaf plants, I can't tell, maybe they're maple trees in the background that have dropped their leaves. they're dropping their leaves because those leaves are not going to be able to handle all the sun energy in the wintertime, right? But, here's what looks to be a spruce or a fir. It's cold outside, it's a sunny day, and that's those two factors, sun and cold, spells trouble if the plants don't have a way to dissipate energy. So, in the summertime, these spruce plant trees are photosynthetically active, their metabolism is ramping up. But in the cold winter months, metabolism slows, and this is when potentially there could be problems with these plants, these trees absorbing excessive energy. So, scientists at the University of Colorado in the beginning of the 1990s, I want to say, elucidated a pathway that helps explain how these spruce trees survive excessive light in the winter months. So they were able to explain how their unique physiology matches their environment. And so these spruce trees, in fact, any type of conifer, has this really elegant pathway of dissipating energy in something that's called non-photochemical quenching. So I'm going to try to break that down. So non-photochemical, so that just means um, the energy that's being absorbed by the chloroplasts in the winter in these trees is not driving photosynthesis. So they're absorbing the energy and then they have to transform it to something else. So normally in the summertime, right, these plants are going to absorb the energy and transform that energy into sugar, right? That's photosynthesis. But because energy can't be just created or destroyed, the plants need a mechanism to transform this energy that is not going to be a part of photosynthesis. So, what happens is that in the winter months, these plants are subjected to photons from the sun, right? They're being absorbed by the chloroplasts, and then that energy is being transformed and released as heat. So it's a really wild mechanism that these, conifers have evolved, right? They're not, because again, they have their leaves, these needles, but they can't drive photosynthesis, but they're sitting ducks, right? And I say that because they can't move, they can't put up an umbrella to shade themselves from the sun. So they're absorbing that sun energy, transforming that energy as heat and releasing it to the environment in a process called non-photochemical um, quenching. Okay. Next, I want to go ahead and talk about some stress that my research program investigated. So what I want to talk about next is metal stress, and the example here is how some plants can deal with excessive nickel. So this plant here is in the genus Alyssum. This photo was taken. I can't remember if I took this in Greece or Turkey, but I had um, I guess the privilege, the opportunity to work in Turkey and Greece, well, Turkey for a year and Greece for 4 summers, to study these plants in their natural environment. So these plants are growing on serpentine soil, which is really, really high in nickel. Um, and so to grow on nickel, these plants they can either do one of two things if they're going to thrive in this in soil that's has very, very high concentrations of nickel. So one option is that these plants have evolved a system to avoid the nickel, right? So maybe their roots and the transporters in those roots can decipher what ion is nickel and what ion is not, right? So maybe one option is exclusion. The second option is accumulation and tolerance. And so in this case, the second option is the correct pathway. So these plants are not avoiding nickel. In fact, they're hyper accumulating it. So they're taking up excessive amounts of nickel from the soil and they're detoxifying it. And so they have a couple of different mechanisms from this, but basically it's all related to your biochemistry. And that biochemistry is determined genetically, right? So they have these genes that can be upregulated in response to nickel, and they're going to produce certain chemicals. Some of these are acid, some of these are, malic or citric acid can bind to nickel and detoxify it. Glutathione is an antioxidant that can bind to nickel. Histidine is an amino acid, and histidine, when it is at high levels, it can also bind to nickel and partially detoxify it. So this is a detoxification strategy. But still, these plants have to, there's so high amounts of free iron floating around these plants. Um, but that's one way that these plants can tolerate very high levels of nickel in their soil. And I should mention that serpentine soils with high levels of nickel are found along the Mediterranean coast, but they're also found in areas of California, and I want to say some areas of China. So it's not as if it's just endemic to Turkey or Greece. Um, Similar to metals, I'll also talk about metalloid stress. So the metalloid that we'll consider here is selenium. Selenium is unique in biology because it is not required by plants. However, it is required by bacteria and mammals. So that's a really unique evolutionary pathway, right? How bacteria, cyanobacteria have a requirement for selenium, somehow skipped plants, right? And then that requirement for selenium still now shows up later in animals. So to the best of our knowledge, every single animal, from humans all the way down to the simplest animals, have a requirement for selenium. That's because selenium in humans and bacteria is used to make the 21st amino acid called selenocysteine, in which the sulfur in cysteine is replaced by selenium. And I like to bring this up, right? Because in biology, we're always taught that there are 20 amino acids. Well, in fact, there are 21 proteinaceous amino acids in humans that are required for our survival. Okay, so back to selenium. So this shown here is a picture of a Stimleia pinnata plant I took in Colorado, some areas of Colorado, Wyoming, California have just naturally rich high levels of selenium. Um, Stimleia pinnata is a plant that has evolved mechanisms to tolerate high levels of selenium. It is also going to be classified as a hyperaccumulator. In other words, so this plant can hyperaccumulate very, very high levels of selenium. Um, we're going to talk about two different approaches that this plant has evolved to survive in selenium-rich soil, and also ways to accumulate high levels of selenium without killing it. So one is volatilization. So in the soil, selenium exists as the ion selenate. Selenate will be taken up by the plants, and it's going to be transformed into selenocysteine, and then it's going to be methylated. So you just tag that selenium compound with a little methyl group, and then it's volatilized as diselenium methyl. Oh, I can't remember the exact chemical. I apologize. But anyways, it is methylated and it's going to be volatilized, meaning that it enters the selenium goes from the root through the xylem into the leaves. It's going to be methylated and then it's going to volatilize, or in other words, escape from the plant as a gas into the atmosphere. And that's pretty rare strategy for how plants can deal with metals or metalloids. So most plants, whether it's iron toxicity or copper or manganese, right? Zinc, right? Those metals cannot be methylated and then volatilized into the atmosphere like selenium. So that's a really special, unique case, explaining how these plants can survive in very rich, selenium-rich soils. Another strategy that these plants have evolved is to sequester the selenium in parts of the plant that are not metabolically important. So there's nice research done showing that selenium can be sequestered in trichomes, and shown trichomes are these tiny little hair-like extensions found in a variety of plants. They're observed in tomato plants. They're often thought to decrease herbivory, and so the plant is able to transport the selenium to these trichomes, right? So get it away, get this toxic selenium away from organelles that are metabolically important, right? Get it away from the nucleus, even though not much metabolism happens in the nucleus. Get it away from the cytoplasm, get it away from mitochondria and chloroplasts where there's lots of metabolism, lots of enzymes that could be interfered with as a result of the selenium. Store in the trichome, and it should decrease herbivory if a plant were to eat these trichomes. I suspect that there might be people out there who've heard of trichomes. Cannabis plants also have trichomes, and so trichomes in these cannabis plants also have elevated levels of the active ingredient in marijuana, THC, right? So THC is another plant secondary metabolite that these plants have evolved the capacity to synthesize, right? That they send this molecule to the trichomes, and it's going to decrease herbivory. In other words, if an insect eats those trichomes on that marijuana plant, it's going to, it's physiologically, it's not going to be able to do well, right? It's that insect might be subject to predation, or it's certainly that insect or animal is not going to function as it should. So that's I guess an overlap between these cannabis plants and this Stanleya pinnata plant, which has evolved on selenium-rich soil. So I gave two examples of how of hyper accumulators. I talked about these nickel hyperaccumulators from the genus Alyssum and selenium hyperaccumulators. But they're found worldwide, right? So there are plants that can accumulate 2% cobalt, 2% zinc. There are plants or ferns in Panama that can accumulate 2% arsenic. So this is a really odd phenomenon, right? Because 2% arsenic, selenium, nickel, zinc, or cobalt would be lethal to all other forms of life, to the best of my knowledge, right? I mean, next time you run across some baby infant formula, look at how low the levels of zinc or selenium are, right? It would be like per serving, it's like 0.00 micrograms, right? So you just need it in a tiny, tiny, tiny amount, right? So it's a trace element. So, but these plants, man, they're just like chock full of these metals and these metalloids that would, kill any other microbe or plant out there, right? So certainly a tomato plant could not have 2% arsenic or 2% nickel. So, that kind of begs the question, what's going on here? Like, why are these plants actively and purposefully trying to accumulate these metals in their environment? I wish I could see the audience, right? Because hopefully I'd see some hands going up. And you'd be right to guess that these plants have evolved this strategy as a defense mechanism, right? So they're accumulating arsenic and nickel to deter herbivores. And there have been lots of these cool experiments done with crickets. If you give cricket a choice between a plant with or without the selenium, they're always going to gravitate to the plants without selenium. You can do it, you can infect the plants with mold, or not mold. Yeah, a mold or a fungus, right? Powdery mildew. And they're always going to grow better on the plant that has not accumulated these selenium or nickel. So you can do both a choice experiment, right? So this is a controlled experiment in a lab right? Where in a container you have, an alyssum plant grown on nickel, an alyssum plant grown without nickel, and these pests are always going to be chomping on the plants that have not grown on nickel. You can also do a non-choice experiment, right? So in this case, you have two plants grown with and without nickel, but now they don't have a choice, right? So now they're sequestered, right? There's a divider between these two plants. Plant one grown with nickel has 100 insects, crickets. There's a divider, another plant grown without nickel, 100 crickets, right? And the mortality of the crickets that do not have a choice, they can either starve to death or take their chances and eat this plant loaded with selenium, nickel, arsenic, right? And they're going to succumb, right? They're just going to just not do well. They're going to lose body weight, and eventually, the mortality rate will be much higher compared to the plants that have not been grown on arsenic. So that's kind of a cool evolutionary response that these plants have evolved. And again, it's seen worldwide. Wherever there's like a very high level of a metal or metalloid, there are always going to be some plants that hyperaccumulate these. So that, when I was in academia, my research program just investigated the molecular, the cellular mechanisms involved in hyperaccumulation, and it extended beyond, I guess, what I'm presenting now. So, if there are questions on it, I'm happy to discuss it at the end or later via email. But I was looking at a system called the proteasome, which can degrade damaged proteins. And so it was very molecular, cellular based, but it was fun. I enjoyed it. So much that I resigned and moved to the gorgeous state of Maine, where I met Jack and later Jeff. All right, I'm going to go ahead and illustrate one more example of stress in the environment, and we're going to talk about nutrient stress. And this is also another project that my research program investigated. Plants are always competing for nutrients, and plants have evolved so many elegant pathways to acquire nutrients and send those nutrients to the right spot. Um, so I'm going to deviate from the slide right now. So the really cool example of this is copper, right? So copper is a required trace element in all organisms. Um, in humans, there are proteins in the mitochondria and the cytosol that require copper. In plants, there are proteins in the mitochondria, chloroplast, and cytosol. And there's competition for copper for the 20 or so proteins that require this essential trace element. But let's imagine a situation where there's a limited copper, right? Let's imagine a situation where the plant has a budget, a copper budget, right? And the plant is being starved of copper, but it has to decide which protein that copper is going to go to, right? And so plants have designed a mechanism to target which protein the copper goes to. And it does this, it's a post-transcriptional modification. So the plant will degrade the mRNAs of proteins of the competing copper requiring protein. So basically, if that mRNA is degraded, the protein will cease to exist or the protein won't be created, which leaves just one copper protein left to which that copper is going to go. So that's just one way that plants in the natural environment, as well as agricultural plants, have evolved to allocate and determine where nutrients inside that plant get to. Um, so this slide looks at possible nutrient stress in the well-known plant Spanish moss. So Spanish moss is a plant that's found, right in coastal Carolina, right? And so it's endemic It's found its geographic distribution is very broad, right? It goes from northern Virginia, maybe southern Virginia, all the way through, North America, and I believe it extends as far south as Uruguay, right? So it has a very broad geographic distribution. This plant does not have true roots, so this plant is an epiphyte. That means it's going to grow on another plant to survive. And so in this case, this picture here, the Spanish moss is growing on this oak tree, and these modified leaves are really, really slender. This plant is not a moss; it's a true plant. Sorry, it's a true higher plant. It's eukaryotic. It has these flowers, so therefore it's a angiosperm. So it produces these really, really delicate flowers. And so, at first glance, it would seem that these plants have it really, really hard, right? Because they have to acquire all their nutrients from atmospheric deposition, right? Right from rainwater or from the leachate higher up in the canopy of that tree. Maybe it's leaching some nutrients down that the Spanish moss can acquire. So I went into this project, with a hypothesis that these plants are going to be limited by nitrogen. Um, and I had this hypothesis because most terrestrial plants are limited by nitrogen. If you give them nitrogen, they're just going to respond positively. They're going to ramp up metabolism. They're going to increase growth. And that's because nitrogen is the most abundant um, element in plants besides, things like oxygen, hydrogen, and carbon. So after those three, nitrogen's the most abundant. And that's because it's nitrogen's used to make amino acids, and then those amino acids will be used to synthesize proteins. So I had a whole bunch of experiments. These plants never responded positively to nitrogen. So, in other words, I would give them nitrogen, their growth rate, their photosynthetic performance, nothing was budging. Nothing was increasing. It was almost as if, they weren't acquiring the nitrogen. So that was weird, right? So, I did some metabolic studies. Let me backtrack. I did some nutrient analysis, and there's a ratio that you can look at in most plants to determine if they're nitrogen starved. And this is the carbon to nitrogen ratio. And in a lot of plants, that carbon to nitrogen ratio is, I don't know, let's call it for simplicity 20 to 1, or maybe it goes up to 30 to 1, right? And if all of a sudden a plant carbon to nitrogen ratio jumps from 30 to 1 to say 35 to 1, that would indicate uh, nitrogen deficiency. Well, these plants had a carbon to nitrogen ratio of 60 to 1. That's crazy, right? I mean, so again, so I use this preliminary data, as some evidence to indicate that these are plants are nitrogen starved, right? But I took a closer look at these plants, and so even though these plants have a very low nitrogen status, they have an excessive amount of amino acids. So that's bizarre, right? So the total nitrogen, sorry. So if you were to look at the protein content in these plants is very identical to other plants, right? But their amino acids are sky high, and that was weird, right? So it's almost as if like those amino acids aren't being used to make proteins, and that's probably the case. So what's happening here is that the plants are dumping excessive nitrogen into one key amino acid. It was asparagine. So asparagine is the one amino acid that it's the amino acid that has the highest nitrogen to carbon ratio, right? So if a plant is experiencing excessive nitrogen and doesn't know what to do with that nitrogen, it's going to dump it into asparagine. And levels of asparagine were through the roof. I mean, I looked at the literature, and there, to the best of my knowledge, there's not a single plant that has levels of asparagine as high as Spanish moss. And so, what this indicates and helps explain, well, it indicates that the plants are not nitrogen starved. And it helps explain why these plants did not respond positively to nitrogen when I gave it to them. So, one thought is that these plants are doing just fine, right? They're not stressed at all. And perhaps that explains their broad geographic distribution, right? They're found from Virginia all the way to Uruguay. They're doing just fine. But what it also might indicate is that these plants have evolved a very unique nitrogen metabolism. Like they're very stingy with their nitrogen. And um, perhaps let me go back and try to explain why they're dumping this nitrogen into amino acids rather than just storing it as nitrate or nitrate. And so when a plant like Spanish moss acquires nitrate, it's going to convert it into ammonium, and that buildup can be toxic, right? So there has to be an ammonium detoxification strategy. The strategy to detoxify excessive ammonium and ammonia is just to convert it into this amino acid. So they're dumping all this excessive nitrogen into this surplus amino acid. It's just staying there. Um, so, you know, so this is a classic example of the data do not support the hypothesis. I'm comfortable with that, right? I don't have to reinvent my hypothesis just because the data don't support it. Perhaps these plants are limited by another element, maybe maybe iron. Perhaps they're limited by the amount of cellular energy, like ATP, that is required for protein synthesis. So protein synthesis is very energetically expensive. And so I really don't have a good explanation, right? That's part of the fun of science, I hope, right? I'm comfortable with, I wouldn't call it failure, but I would certainly, give this as one of many examples of experiments that I've done where, the data just do not support the hypothesis. And that's part of the fun part of science, right? So this did not deter me. Unfortunately, I never got around to publishing this research on Spanish moss. Life happened. I moved to Maine. I resigned from the university, and all is good. But I think it's a cool story of how these plants that were just around campus. Everyone thinks they're nitrogen starved. You give it nitrogen, and oh my goodness, these plants are just gonna like just totally take control, right? And so people would speculate, oh, perhaps climate change and increased nitrogen status and acquisition. You'll have these Spanish moss plants growing all over the place. But in this case, I don't think people have to fret about Spanish moss dominating and taking over, habitat and growing everywhere in South Carolina. So folks, that's all I had. But I'm happy to, entertain any questions if you have them.
Wow, thanks, Doug.
Yeah,
That was great. I knew it'd be exciting, but that's a great introduction. Looks like we've got at least one hand raised already. If you want to go ahead and, unless those are just claps. It's like Annie has your hand raised, maybe? Can you see
Uh,
the chat, Doug, or do you want me to read these out?
yeah, can you please read it out? I saw the chat, then it just disappeared. Oh, hold on.
Sure,
Yeah,
sure,
I see it. I can't. Yeah, can you please read it out, Jeff?
sure. Lydia is asking, in the experiment with the alyssum grown with and without nickel, was there a noticeable difference in the plant health slash morphology slash germination, or is the alyssum adapted to high nickel? Sorry, I've got something weird happening with my screen. Um, where there's no measurable negative impact. She's curious if there is a trade-off or downside to the nitrogen accumulation scheme.
Yeah, great question. So no, the plants do not have a requirement for excessive nickel. There's no discernible difference between these plants. So if you grow these plants in the absence of stress, right, these plants do just fine without, high levels of nickel. So visually, we would not be able to detect which one has nickel and which one does not. But if you give them nickel, the plants are going to
Some
take it
follow-up,
up.
but looks like Alyssa. So Alyssa also has a question.
Yeah, hi. So I also had a question about the hyperaccumulators, mostly just what you usually expect ecology-wise, because it's an amazing adaptation, but I'm thinking if these heavy metals are sort of accumulated in their tissues and they those off herbivores, how does that work ecologically? Like, do the ecosystems look different where there are just high levels of heavy metals in the soil?
Yeah. So the soils have high levels of heavy metals, and mostly, that's just determined by the bedrock that's underneath, the soil and those plants. But but I'm afraid, Alyssa, I'm not answering your question. Can you state that again?
Yeah, yeah. I don't think I said it clearly, but just I was thinking like how are, you know, not all herbivores dying off in this particular ecosystem? Like how is there still, you still have like a functioning ecosystem even in these environments?
Yeah, that's okay. That's a good point. Yeah, so I think I understand now. So in soils with very high levels of nickel or selenium, you do have plants that are not selenium hyperaccumulators. And
Mm,
sometimes those plants are doing fine, right? Sometimes that's not the case. So certainly these plants have an evolutionary advantage, right? Uh, the Yeah, so it's not as if it's a monoculture. It's not of this as if that it's,
right.
although these plants are the dominant species, they're not all nickel or selenium hyperaccumulators. So there are some other plants that are out there.
Okay. That makes total sense. Thank you.
Yeah, you're welcome.
Any other
Good question.
questions? I guess it's my bias by coming through. Any genetics nerds out here are probably familiar with Thermus aquaticus in terms of, I'm not sure if extremophile is an appropriate term anymore, but if your DNA replication amplification, that was one of the bacteria amplified
Yeah,
the hot springs in Yellowstone. That became a huge issue legally as well, but really helped launch an entire just massive wave of applications for conservation in large part. Um, yeah, that's more what I'm familiar with. This is a great addition.
yeah, Jeff, I'm glad you brought that up, because I use that example to illustrate why basic scientific research should be maintained, right? We never know what we're going to discover when we do this basic research, right? So Jeff was talking about, isolating the DNA from bacteria from these hot sulfur springs where the temperature is like 90 degrees. So these are
Celsius.
thermophile. Yeah, and right. And so from that, weren't they able to then isolate the DNA polymerase that's used in PCR from that?
Yeah.
And yeah, a
Yeah, it
whole
launched
bunch.
a multi-billion-dollar industry essentially. Then that's where the fights came in into who owned that because it was located on federal property, national property. But yeah, it allows amplification much more quickly
Yeah,
than what our cells were have evolved to replicate in. Yeah, it's changed. It was
right.
an absolute game changer.
Yeah,
Yeah,
absolutely. And it's a great example of how you never know what basic research, how it can be translated to something, magnificent.
it is. If folks interested, there's a great, very short, non-technical article about I forget the birth of PCR, something like that. The guy who actually put all the pieces together on a drive through the Sierras, as I recall.
Cool.
I won't go into that anymore. If folks have any more questions, feel free to speak up, or like Doug said, feel free to reach out to him anytime. Here is a link, in case you haven't seen it, to his course that's beginning here in, like I said, a little bit less than a month. Uh, this is a great plug for why we all need to increase understanding of these adaptations and how they can have downstream effects and what they can tell us about these communities that we're working in. Also put in a link to our playlist. This recording and others will be this will be up in a few days, given how I tend to operate and getting them posted. But we do have our full library up there currently. And to put in also one last plug for this particular program, we'll be having another presentation in a month, and I will be co-hosting that one. And it's similar to what Doug's been saying. If you're into potentially debunking some popularly held myths that seem to hold on in science, that's going to be the discussion about some certain bear behaviors, literature review that I'm helping with. So if you're into bears at all, that might be of interest. As always, if folks have topics they'd like us to discuss, any ideas for other courses we could provide, or if anyone's interested in working with us, any of our staff, instructors, or so on, please reach out anytime. It's great to hear from folks. And it looks like no more questions. So with that, I will give one last thank you to everyone who showed up today. We love to see these attended, and again, share the recordings with your colleagues. And a huge thank, of course, to Doug for the extra time put into this. It was a fantastic presentation. I'm super happy the timing worked out. And I will be trying to sneak into your class if they let me here in a month.
Thanks, Jeff. Thanks, everyone.